Good evening, students. Welcome back to Sanita Chatterjee's biology classes. Today, I will be discussing two very important questions that are not only essential for the upcoming JENPAS-UG 2025 examination but also highly relevant for competitive exams like NET and GATE in life science. These concepts form the core of molecular biology and are frequently asked in various entrance and postgraduate level exams also. Let's solve the questions. So, here is the question that I have selected. It has come on JENPAS UG 2024, very recent, and firstly we will see the question, that is cDNA is copied from mRNA with the help of which of the following? Okay, so the first tool to solve such type of problem is read the question thoroughly before trying, or before, do not panic and do not think too much. Okay, so the whatever the very little information that has given to you, firstly extract the whole amount of information that is known to you, like cDNA. What is cDNA? Cdna means complementary DNA. Cdna means complementary DNA. Complementary DNA. Now, do you know what is complementary DNA? Complementary strand, you know. So DNA is made up of two strands, like two strands twisted into a double helix, right? So a complementary strands refer to the second strand of a DNA molecule. See, if it is a DNA molecule consists of two strands, it's one strand, and there must be another strand like this way, right? So this one is the complementary strand. And how this complementary strand formed? On base pairing rules. Okay, so what is the base pairing rule? Like, if this strand is adenine, then there must be thymine. Okay, so this is the complementary DNA strand. Now we need to make a complementary DNA is copied from mRNA. We know that mRNA is a single stranded. So if it is a mRNA is single stranded, right? I am preparing a complementary strand of this mRNA, that means a complementary DNA, because if there is two strand, then it is not RNA, it will be DNA, right? So which enzyme helps to form this cDNA from this mRNA? That is the question. Now, in molecular biology, these enzymes are really very important, and they play a very crucial role, right? And they are specific for treating specific type of equation. Restriction enzymes, reverse transcriptase, DNA polymerase, topoisomerase, they all play separate important role. And maybe someone is playing role in replication, someone is playing role in transcription. Okay, we don't know, but to solve this question, we must know the functions of the enzymes that are responsible for replication, transcription, and translation. So in the next slide, we will see the answer. So the answer is reverse transcriptase, option B. Why? What is the function of reverse transcriptase? Reverse transcriptase used to study gene expression by converting RNA into DNA. So this enzyme is responsible for a particular thing, that is reverse transcription, PCR. Normal PCR means to amplify DNA, but RT-PCR means to copy, to amplify the cDNA from the RNA. Okay, that is the difference between RT-PCR and this is regarded as RT-PCR. For RT-PCR we need reverse transcriptase. Okay, and we form RNA into DNA. Then what happens? Regular PCR steps with DNA polymerase to amplify the cDNA. We know that PCR means polymerase chain reaction. PCR means polymerase chain reaction. Why this technique used? This technique used to amplify the DNA? Why we need to amplify DNA? That means multiplication of DNA for our gene, studying the gene expression, for cloning, for making copies of DNA. We need polymerase chain reaction, that is PCR. But RT-PCR stands for reverse transcriptase PCR. Okay, so here is the RNA strand, and see we are preparing a complementary cDNA, these strands. Okay, so now we will see why the other options are not correct. Other options are, so the option B is correct, and these A, C and D, why they are not correct, we need to know. See, so firstly again, reverse transcriptase. Okay, reverse transcriptase is an enzyme that synthesizes complementary DNA, that is the cDNA, from an mRNA template, and this process is essential in molecular biology for studying gene expression, cloning eukaryotic genes in prokaryotes, and creating cDNA libraries. Okay, and the others, why they are not selected? Because restriction enzymes, they cut DNA at specific sequences. That means it splits DNA at specific sequences but not involved in copying mRNA to cDNA. Okay, and then next, that is DNA polymerase. This enzyme is responsible for amplifying the DNA for making copies of DNA, that means DNA synthesis. DNA from a DNA, that means replication, but not from RNA. Okay, DNA forms from a DNA template, not an RNA template. Okay, and topoisomerase, it relieves supercoiling in DNA during replication. That means topoisomerase is also used in DNA replication process. That is why I told you that you must know the functions of enzymes that are required for replication, transcription and translation. Okay, next, move to the next question. See another question from JENPAS-UG 2021 on this similar context. The enzyme used in transcription is, firstly I have told you that you need to read, you need to study these three types of process, that is replication, transcription and translation. That central dogma rule, that is DNA to RNA to protein, DNA to RNA to protein, right? So DNA to DNA synthesis, that is the replication. DNA to RNA is transcription, and RNA to protein is translation. So here is the question asked from transcription process. So before going to discuss the rule is same, that you must know the enzyme that are required for transcription. If you know the functions of transcription, then it will be very easy to select the correct option. So there are options: DNA polymerase, ligase, RNA polymerase, and transcription and transcriptase. So see, JENPAS-UG 2021, and the enzyme used in transcription is, the answer is C, DNA polymerase. Okay. What is the explanation? Transcription is the process of synthesizing RNA from a DNA. Okay. So DNA to RNA is known as DNA to RNA. Here occurs the transcription. Here occurs the transcription. Now what is the enzyme? That is RNA polymerase, option C. And RNA polymerase, for prokaryotes, only one type of RNA polymerase can make these three type of RNA, like mRNA, tRNA and rRNA. To synthesize all type of RNA, we only required a single type of RNA polymerase. But for eukaryotes, okay, for eukaryotes, there is three type of RNA polymerase. RNA polymerase 1, that makes rRNA, RNA polymerase 2, that makes mRNA, and RNA polymerase 3, that makes tRNA. So this is another important part. Remember these things, this information, that RNA pol 1, what is the function of RNA pol 1, RNA pol 2, RNA pol 3. Maybe this time the question comes from this part, that RNA polymerase 1 is responsible for making or synthesizing which type of RNA, that is rRNA. Okay. And the roles of the other enzymes, why they are not correct? Because DNA polymerase synthesizes DNA during DNA replication. We already know this information from the previously solved question, right? And the ligase, the new enzyme, it joins DNA fragment. Okay, Okazaki fragments during replication. And transcriptase, not a standard term, sometimes used informally for reverse transcriptase, which is unrelated to regular transcription, that is not related to transcription. So the left one is the C, that is the RNA polymerase, and that is the correct answer. Okay. And see, this is the role of RNA polymerase. This is the enzyme RNA polymerase. RNA polymerase reads one of the DNA strands, these strands, and adds complementary nucleotide. Like if it is A, it will be T. Okay. If it is G, it will be C. So guanine, cytosine, adenine, thymine, complementary nucleotide bases to make an mRNA transcript. This is the newly synthesized mRNA. Okay, that is synthesizes from the DNA. This is DNA because it has two strands. From DNA we are preparing RNA. That is why this process is transcription, and this enzyme is RNA polymerase. And for eukaryotes, RNA polymerase 1 prepare rRNA, RNA polymerase 2 prepare mRNA, and RNA polymerase 3 makes tRNA. Okay, remember this information. So hope this video will be helpful for your preparation. So stay connected. Bye bye for now.